I think in the interest of time, we will start off. Welcome everyone to the 2023 Team FDMAS Research Update. I'm Tova Burstein, Community Engagement Director with the FDMAS Alliance, and I'll be going over some quick announcements before we get started. Today's program is being recorded and will be available online afterwards. We ask that you keep yourself muted and off video until the Q&A portion at the end. We will be pausing for questions between presentations, and you can ask those directly or put them in the chat to the group or to Dr. Collins, our moderator for today. Today's program is made possible by the entire FDMAS community, but I want to recognize in particular two individuals. The first is Lily Lagno of Association MASFD in France. Lily single-handedly funded one of the projects we'll hear about today, that of Dr. Ted Abel. She started MASFD and Supportive Skeins, her organization, which sells yarn and knitting patterns out of love and devotion for her daughter, Gaia, who is deeply affected by this disease. Lily, what she's managed to do is incredible, and the entire FDMAS community is humbled and deeply grateful for your work. And just very inspired by what she's been able to do. I'm also honored to tell the story of our Vice President, Cindy Brandt-Levin. Cindy has served as team captain for Team FDMAS for 9 years now, and similar to Lily, she started our involvement in Team FDMAS out of love for her daughter, Carly, who has this disease. As a parent, she was frustrated that there weren't more ways to support and encourage research in FDMAS. And she went and found the UPenn Orphan Disease Center $1 Million Bike Ride, which we've been involved in for this is our 9th year now. And since 2014, she's led our community to raise over $1 million and supported 18 research projects focused on FDMAS research, which is incredible, but that number, I think, doesn't truly capture the lasting impact and the research collaborations that have sprung from these projects and webinars like this one. Just for example, this year, Dr. Yingzi Yang, a previous Team FDMAS awardee, was recommended for over $3 million in funding through a Department of Defense peer-reviewed medical research program. Truly, I'm blown away by the progress this community has supported, and we could not have done that without Cindy's dedication and leadership. Today we're celebrating the research funded in 2020, but we're also in the very beginning of our 2023 campaign. Team FDMAS is lucky enough to have a very generous match gift from the Cohen family of $30,000. And so that gift, along with the UPenn match of $30,000, means that all gifts right now for FDMAS research are tripled. I will put the link to our campaign in the chat later. For Gaia, for Carly, and the whole FDMAS community, we really want to see this progress continue. We're very grateful to the scientists who are sharing today, and we hope in 2023 we're able to fund even more brilliant projects focused on understanding and treating FDMAS. With that, I am honored to turn it over to Dr. Mike Collins. Dr. Collins is senior investigator in the Skeletal Disorders and Mineral Homeostasis section of the National Institute of Dental and Craniofacial Research, and he's devoted his career to the study of fibrous dysplasia, McCune-Albright syndrome, and rare metabolic bone disease. He's maintained close collaboration with FDMAS Alliance since our founding and served as our medical advisory chair for over a decade, and is currently our scientific advisory chair. Mike, we're so grateful for all that you do, and thank you so much.
Well, thank you, Tova, for that introduction. And again, thank you to the Brandts, the Cohens, and Lilies for the amazing work they've done to raise funds for this. And as you said, it was a grant for Lily, who was specifically interested about the neurocognitive aspects of McCune-Albright syndrome, that directed this grant to Dr. Abel, who will speak first. Dr. Abel is director of the Iowa Neurosciences Institute at the University of Iowa. He's a professor there, chair and department executive officer of neurosciences. His laboratory focuses on the molecular mechanisms of memory storage and the molecular basis of neurodevelopmental and psychiatric disorders. And I'll stop there and just turn it over to you, Ted.
Great, Mike, thank you, everyone. It's really terrific to be here. And what I want to do today is really tell you about our findings over the last couple of years, which have been very exciting. And so let me start by sharing my screen. So with me today is Satya Tadanada, who's a postdoctoral fellow in my lab, who has been working on this project and really is responsible for the data I'll show you. And the support from Lily and the foundation have really made his postdoctoral fellowship possible in the lab. Thanks everybody. Let me see if I can get my screen shared right here. How does that look okay in terms of seeing my slides, Mike?
Perfect.
Yeah. So what I want to talk about are, thanks, Tova. What I want to talk about today are the CNS. And please, if Mike or Tova could keep an eye on the chat, or if people want to raise their hands and if you guys want to jump in, I'm happy to have questions at any point. It is no problem. I'm going to be talking about things pretty scientifically. So, but I'm happy to explain them more generally, and I'll try to do that as we go through. So I'll talk about CNS manifestations of McEwen-Albright syndrome, beginning with some clinical work at NIH and then talking about our work in mice. But first, I want to thank, as we did earlier, Lily and her supportive SKEEN initiative with the cells donated knitting patterns and wools to support FDMAS research. This is the announcement from in Iowa Now, one of our magazines here, when our grant started in 2020, announcing this award. And so for Lily and Gaia, this is for you, and thank you so much. We have a son, Seamus, who's now a man, 23 years old, on the autism spectrum, and so I've lived the life of a parent whose family is impacted by a serious disorder. I was involved in a number of initiatives in autism. One of them was a foundation that started what was called the Autism Genetics Resource Exchange in the early 2000s. And that was the first large-scale genetic database that gave rise and connected with what's now going on at Autism Speaks and at the Simons Foundation to really have these, and a thing called Spark, which has, I think, over 100,000 family individuals in it that include families of individuals with autism. So I've seen firsthand the impact that parents and family can have to help make a difference in understanding disease. And I hope that maybe in what we're talking about might actually reveal something interesting about McEwen-Albright syndrome. I think it actually has. It's a genetically complicated thing we found, but I think you'll find it to be very interesting. So So we all know about McEwen-Albright syndrome. I really don't need to tell this group that, but one of the challenges, firstly, just genetically, I'm going to focus on the genetics, this mutation called R201H, that is a gain of function in G-alpha-S, GNAS, and that leads to a hyperactive signaling with GTPase activity. This mutation, as you guys know, occurs somatically during development, and then individuals with MAS are mosaics for this mutation. Now, we got into this and really connected with this from my work in memory consolidation. And my lab works on the role of cyclic AMP signaling in memory consolidation. And I was a postdoc with Eric Kandel at Columbia University College of Physicians and Surgeons, where we made some mice that looked at protein kinase A signaling. When I started my own lab at the University of Pennsylvania at that time, I made a number of mice to manipulate cyclic AMP signaling. One of them was a constitutively active G-alpha-S, a different mutation from the one that's in many of the MAS cases. And in transgenically overexpressing that, which at the time in the 2000s was the approach we had, Mishi Kelly in my lab found some very interesting deficits in memory, which we think would reflect some alterations in hippocampal function. So it's about context and space. Deficits in a task that I'm going to talk about called prepulse inhibition, which is called sensory motor gating, and structural abnormalities with enlarged ventricles and thinned neocortex. Now, we went on to show that particularly these features of pre-pulse inhibition, that measure, which I'll describe later, is used as a preclinical model for efficacy of antipsychotics. And so while we can't have a mouse that is psychotic, we can test drugs that in humans treat psychosis in mice. And they target these particular behavior called pre-pulse inhibition. And so what happened, I guess now about 7 years ago or so, or maybe even longer, a psychiatrist at Yale was seeing a patient with McCune-Albright syndrome that had many symptoms that she thought were symptoms of psychosis. And she looked through the literature and she found our paper in molecular psychiatry in 2009, connecting G-alpha-S signaling to at least an animal model of aspects of antipsychotic efficacy, which is connected, we think, to psychosis. And so that got her thinking about that. And we contacted NIH and people at NIMH, as well as Mike Collins, to think about this in MAS patients. And this group here, sorry, I'm having trouble with the various subscreens on my screen here. So this group here, led by Dr. Joyce Chung at NIMH, and then Michael Collins and Allison Boyce, looked at patients that came to NIH, and they were interviewed and screened and seen by a psychiatrist at the National Institute of Mental Health. And what they found was an increased risk of psychiatric abnormalities in individuals with McCune-Albright syndrome. And that included disorders like anxiety disorder and a mood disorder and ADHD, as well as psychosis. They then looked at whether this was related to endocrinopathy and found that it did not seem to be directly related to endocrinopathy. That's something we sort of thought it might be connected to that, but it seems like it's not, especially psychosis. And so this work has been presented in abstract form. The group need to put this together as a manuscript, but it means that for individuals with McCune-Albright syndrome, in particular, as they age, may develop and need to be followed by clinicians with more of a focus on psychiatric conditions. There's some biochemical studies that in individuals with schizophrenia and bipolar disorder, bipolar disorder also includes psychosis as a feature in some patients, that there's increased G-alpha-S signaling in post-mortem tissue from those subjects. So this is an interesting area of research, and what we have looked at here is to sorry, I'm missing my slides a little bit. We looked at a way to make a mouse that would manipulate this pathway. But first, I want to highlight something, because we're going to wind up talking about something that's going to be a little bit complicated. I think some of you may be familiar with it from Lee Weinstein's work and others. And that is that the G-alpha-S locus is very complex. It has a number of mRNA transcripts, both sense and antisense, and those give rise to a number of different proteins. And we know that one disorder, Alport hereditary osteoid dystrophy, comes from inactivation of one copy of GNAS. Now, these in the human genome, there are 2 copies of each chromosome, one from your mother and one from your father, referred to as maternal and paternal chromosomes. And there's been work over the decades that has shown by geneticists that these maternal and paternal chromosomes are not equivalent. And going through the process to make sperm or the process to make egg changes not necessarily the genetic sequence, but the epigenetic modifications of chromosomes that can regulate specific gene expression. So what that means is we have this phenomenon of what is called imprinting, which is that the gene that comes from the mother may be differentially expressed from the gene that comes from the father. And in G-alpha-S, we have that clearly, where there are transcripts made from the maternal chromosome and other transcripts made from the paternal chromosome. And in AHO, that disease results from inactivation of specifically the maternal allele. And so this concept of imprinting means in genetics, we need to think about which copy of the chromosome has this mutation. And we took that into account in our mouse studies, which I'll tell you about in a second. So to study this in mice, we used a mouse line developed at Harvard and the Khan et al. A PNAS paper, and it's a so-called knock-in mutation of this R201H, and it's designed in such a way that it is conditional. This is from Dr. Yang's lab at Harvard, and what she did was to make an allele of G alpha S that has two sites on it. These are called LOXP sites. What that means is that they can be targeted by an enzyme called a recombinase, which is Cre recombinase, that we can express in any cells in the body pretty much that we would like to. We chose to express this recombinase in neurons so we could look at the CNS effect. And using a model called an EMX Cre mouse line, basically it's a mouse line that will delete anything that has these LOXP sites on it in neurons that are in the forebrain of the mouse and throughout their development. And when Cre acts on this LOXP sites, it changes this GNAS locus from a wild type locus to a locus that carries the R201H mutation. So in genetic terminology, this is a conditional mutation that we can make in one copy of the genome in one chromosome, and that we can make in cells where we target the recombinase. By we, by the way, I mean Satya, in fact. So now I talked about imprinting, and this is where the genetics gets complicated. And I have to say, if you guys get confused, a lot of geneticists are confused about imprinting as well, and I will do my best to not confuse myself. So we have to label our mice as to whether they carry the floxed allele, this allele that's going to give rise to the R201H mutation in the paternal chromosome or in the maternal chromosome. We mate them to these EMX Cre mice, and we generate animals that carry a Cre transgene and carry a modified GNAS locus on the paternal or maternal allele. And so this isn't exactly mosaic in the sense that McCune-Albright syndrome is, but it is mosaic in a sense that only a subset of cells in the body have this mutation, those cells that are going to give rise to neurons in what's called the forebrain. Now, we did a large body of studies with these animals. I'll highlight some of that work. Firstly, is a change in body weight because of the phenotype and AHO and metabolic effects that Lee Weinstein has described in G alpha S mutant mice. We looked at body weights and found that males and female mice. So the other variable we have here is the sex of the animal being studied. So we studied male and female mice carrying either the paternal allele as an R201H or the maternal allele of R201H. In all cases here, animals carrying R201H exhibit lower body weights. So we see that here looking kind of across what is essentially adolescence in a mouse from 6 to 10 weeks, becoming adults, late adolescence. So we did a number of behavioral studies. I'll highlight some of them here. We looked at fear conditioning to look at memory, and found what was sort of a surprising finding to us, that there was an increase in contextual fear conditioning only in females, and only in those females that had a mutant allele that came from their father. And this increase in fear may very well be related to anxiety, and that's seen in some of the McCune-Albright's patients. We think about this as memory, but it's also a kind of fear memory, which could relate to kinds of anxiety, in part because we see it at baseline. As I mentioned, we're particularly interested in looking at sensory motor gating and startle responses. So Satya did that by using an apparatus to measure the behavior of animals. And in this, what he did was present a tone to animals. This is a 120 decibel tone. This is like putting your head in the speaker at a rock concert. It's a loud tone. It's very short, but it's a very loud tone. And the animals jump, and we measure their jump. And that's called startle. And what we found was that the mutant males and the mutant females, when they had the maternal allele mutated, had an increase in startle. And so this also might connect to some aspects of the psychiatric disorders exhibited in terms of also anxiety and mood disorders. We then went on to look at pre-pulse inhibition. Now, pre-pulse inhibition is this measure that is used in studies of antipsychotic drugs, particularly those targeting dopamine. And in this, what you do is this is the startle I talked about on the left, a loud tone, the animals jump. If you play a softer tone, which is about 70 or 80 decibels, a more reasonably sound tone, the animals essentially know that this second stimulus is coming, and so they exhibit a reduced startle. That is called pre-pulse inhibition of startle response, or PPI. This whole thing is called sensory motor gating. And if you think about this, if an individual with deficits in sensory motor gating might have altered reactivity to environmental stimuli. And so what we see in this experiment is, again, the animals receiving the mutation from the mother, these maternal GNAS mutants, have reductions in pre-pulse inhibition, similar, in fact, to what we saw in the G-alpha-S transgenics that I started this talk with. And so this is potentially a measure in animals of something that might relate to, again, aspects of psychotic and CNS symptoms in individuals with MAS. Now, we tried some initial drug treatments with an old antipsychotic, a classic antipsychotic called haloperidol or Haldol, and it targets D2 dopaminergic receptors. And in the G-alpha-S mice that I studied in my lab some time ago, we were able to rescue the deficits in pre-pulse inhibition. But interestingly enough, by the way, not the deficits in cognition. We've tried that initial experiment in the GNAS, the maternal GNAS mutant mice, and have not seen a rescue experiment, and not seen rescue, but we're looking at other ways to do this. This is a single injection of haloperidol, and obviously, in the clinical setting, these drugs are given chronically. And so Satya is breeding more animals to do this experiment with chronic administration. So what have I told you? A whole bunch of data, but it's summarized in 3 things. GNAS R201H mutants in neurons show reduction in body weight. The female mice that express the GNAS mutation from the paternal allele show enhancements in fear memory and fear responsiveness. And those animals, males or females, carrying a mutation from the mother, maternal inheritance, have deficits in sensory motor gating and pre-pulse inhibition and enhanced startle, all of which might relate to what might show up as psychiatric symptoms in human subjects. So like all good science, I think our findings ask more questions than they answer. We have a number of behavioral studies we'd like to look at, looking at working memory and social interaction and other ways to look at startle and startle responsiveness in the GNAS R201H mice. We are beginning to look at brain structure to see if they have changes in lateral ventricles and cortical and hippocampal thickness. And we can also do a whole bunch of experiments now by putting Cre recombinase in various different circuits to see where expression is important. A harder challenge is to try to actually do this mosaically. That's something that would be a really interesting challenge. Now, in human subjects, there's a number of studies that we could think about doing, and that is acoustic startle and sensory motor gating are really done very routinely in human subjects. There's a lab at NIMH that does that, and it can be done at any developmental age and is something that we might really want to think about in McCune-Albright's patients going forward. And then thinking about if there's a way to analyze the parent of the chromosome on which the mosaic R201H mutation occurs. That may be a very interesting thing to look at and see how that relates to symptoms and the phenotypes exhibited and symptoms observed in the human subjects. So let me finish by thanking the folks made this possible. Satya here, not shown here because he's behind me, and the discussions and ongoing discussions. Andy got us all started here when he was more heavily involved. And then the discussions have been really terrific with Mike and Allison and Lee and Joyce as well. We're grateful to Yingzi Yang for supplying the mice to us and enabling us to work with them. And two other folks in my lab, Junko Kasoya, who's my lab manager, and Marisol Laufer, who is our technician in our behavioral core, here wearing her official Iowa neuroscience hat, actually. So thank you very much, and thank you particularly to Lily for making this possible. Feel free to contact me. My email address is ted-abel@uiowa.edu. It's ABEL, which is a little bit different, and you might spell it if you just start typing. And I'm on Twitter at @tedabelneuro, and always happy to have followers. Hopefully we have some time to trying to stop share. Anyway, hopefully you have some time to answer some questions, and I'll try to figure out how I can get unshared.
I think we have time for a couple questions, no, Tova? But before we start with the questions, Lily, who wasn't here in the beginning, has joined us, I see, and we just wanted to thank you again for your efforts, Lily. It was your hard work that made this possible, and thanks again. So Yingzi has a question.
Yes, Yingzi, thank you for joining us. I didn't see the participant list. That's great.
I was a little late.
No problem.
Yeah.
So tell
Nice
me what I said that was wrong, because you know much more about these mice than we do.
Thank you for this nice presentation. You know, I'm happy that you can make a good use of this mouse line. My question is that, you know, is there any connection between the neuropsychiatric defect of this mice and the bone problem? So, I mean, the real question is that, have you looked at the bone of this mice?
So we haven't looked at the bone, but they're not, you know, they're able to do all sorts of behavioral tasks. So they don't have any thing that would be overtly obvious. Mm-hmm. The expression EMX-CRE is expressed in neurons. It's expressed in some neurons that are enteric, if you look at the, you know, and it's also expressed in the brain. So, but we have not looked at the bone. I'd be a little surprised. I guess it would have to be we're also not really in the pituitary or the hypothalamus exactly. I'd be a little surprised about how we would get to bone, but we have mice. Be great if you could maybe send me an email about suggesting what would be a good assay to look at. We're happy to do that. That's a great idea. We also have not looked at other endocrine measurements, which is the other thing that would probably be interesting. And Satya continues to believe that there are 5 neurons in the hypothalamus targeted by CRE recombinase, which he has yet to demonstrate to me. But he's always saying, what about those 5 neurons in the hypothalamus that are actually regulating the body weight? So we need to go, or he needs to go looking for them. I'm not going to, but that's another possibility, is that the EMX-CRE is doing something that is a little bit unexpected. Would you expect to see bone differences?
Yeah, I don't know, because it's forebrain, like you have said. If I actually also don't know whether EMX-CRE is expressed in the hypothalamus.
I'm getting past the note, so Satya can answer the question. He's passing me a note. Answer the question.
Yeah. So if you have done micro CT scan, or you have looked at some markers in the serum, and you know, you probably will have some idea whether there is any bone problem.
But he had a behavioral answer.
So we've done rotor-rod measurements on these animals. I mean, forced locomotion was comparable, and even the movement in open field tests and everything was comparable too. So
what he's talking about is a rotating rod where it gets faster and faster, and the animals have to keep up. So anyway, so they're pretty good in terms of motor control. But we have a micro CT in the basement. We should use it. Yeah, excellent. Okay.
Thank you.
So Julia, you have a question?
I was just curious to know what you think the origin of the weight loss is, whether that it's related to the anxiety and the kind of psychiatric phenotype, or whether there's something hormonal going on.
Yeah, that's a great question. And honestly, it's a very interesting question. Researchers are pretty good at making mice obese. We are less good at making them lean. And so, in fact, it's something to think about and pay attention to. We have another lean mouse that's a neurodevelopmental model, 16p11.2 Dell CND. They are hyperactive. So they lose weight because they run around. These animals aren't hyperactive. So I think it has something to do, we haven't measured food consumption, have we?
Yeah, I was just gonna ask whether
I don't
you've
know
done
if-
paired
I don't-
feeding or,
Yeah, we need to put them in a clamps thing. They're upstairs. We need to move the mice around our building. We got a micro CT downstairs. We got clamps 2 floors up. So we need to do both of those things. It's not, what I can tell you is it's not hyperactivity. It's either food intake or something altered in how they, in their metabolism. And Satya can look for the 5 neurons in the hypothalamus.
But it fits nicely, with the loss of function phenotype of the mice.
It does, which is a little maybe interesting or odd. I don't know.
Fernando, and then we probably we'll have to move on in just 1 or 2 minutes.
Thank you. Very quick, I'm curious about what you said about the maternal versus paternal mutant GNAS. And I wonder how you think that works, because GS alpha is still expressed either way, right? So do you think it's affecting perhaps some other proteins of that locus? Or is it that the GS alpha is actually not expressed at the same level when it's maternal versus paternal? Right. What's your theory?
So those are both possibilities. One thing that's a little bit of a concern genetically, and actually Yingzi might be able to follow up with me over email and with this, is when you do these genetic manipulations, sometimes the genetic shrapnel has a little bit of an effect on expression. And so that's one possibility. We've certainly seen that in some of our models, other models. So it could be expression of GFS, but then why that would be just maternal or paternal chromosome is actually something to think about. The other possibility is I believe the NESP55, which is the other locus that's maternal specific, that doesn't have this domain in it, I don't think. So I'm not sure. It would have to be a different, it's either different levels or maybe there are other isoforms that we don't know about. We need to powwow with Lee, frankly. Thanks for the question, Fernando. I don't have a clear answer to it.
So I think in the interest of time, we'll move on to Mara Riminucci's talk. Thank you very much, Ted and Sasha. That was really nice. And as you said, a lot of questions raised, but there are some ways to answer some of those. So that should be good. And hopefully the research will continue. And hopefully Sasha will get his PhD and get a grant and continue this work.
He's a postdoc, actually. So
through this postdoc
faculty position.
Sorry. All right. So next, we have Dr. Mara Riminucci. Mara is a professor in the Department of Molecular Medicine at La Sapienza University in Rome. Her laboratory focuses on bone biology. She and her late mentor, Paulo Bianco, were pioneers in the study of fibrous dysplasia McCune-Albright syndrome. And she has been the recipient of 6 FDMAS research awards and continues to be very active in the field and publishing important work, and is an active member as well of the International Consortium of FDMAS research. And with that, I'll stop and turn it over to Mara.
Thank you, Mike. Let me share my okay. So can you see it? Am I sharing my?
Yeah, looks good. Looks good.
Looks great.
Okay. Once again, thank you, Mike, and thanks for giving us the opportunity to present our work to our colleagues and to the FDMAS community. Part of the work they have supported in our lab. I'm going to talk about the project that was awarded in 2021. Here is the title. It deals with osteoclast and pain in fibrous dysplasia. But before talking about the project, I would like to remark that this project and these studies are part of a more general line of research that is currently ongoing in our lab, and is focused on the role of osteoclast in fibrous dysplasia. Now, this line of research is based on a very simple consideration. We all know that FD lesions are fibrous osseous lesions, osteogenic lesions, because they are made by poorly differentiated osteoprogenitor cells that expand and produce pathological bone due to the mutation. However, we have to keep in mind that the bone is rigid, is a mineralized organ. So in order for these fibroosseous lesions to grow, someone else has to make room, meaning has to resorb the existing mineralized bone. And there is only one cell type that can do this in our skeleton, and it's the osteoclast. So osteoclasts, as you certainly know, are those cells that continuously resorb, destroy small amounts of bone to ensure skeletal growth, shaping, regeneration. So their function is really critical and must be tightly regulated. And fibrous dysplasia lesions are always abnormally enriched in osteoclasts. And this is because mutated osteoprogenitor cells do produce very high levels of the most powerful osteoclastogenic factor, that is rank ligand. And so osteoclasts are normally found within FD lesions at the growing age, within the fibrotic tissue, and in close contact with the newly formed bone, which is continuously produced and destroyed. And so based on this, a few years ago, we started to investigate the effect of inhibition of osteoclast formation and or function within FD lesions. And this line of research was established and developed entirely with the support of the FD-MAS community. For example, in one of the first awards that we received, we showed that in a mouse model of human fibrous dysplasia, inhibition of rank ligand by using an anti-mouse rank ligand antibody, meaning a murine equivalent of denosumab, it's possible to convert fibrosseous lesions into hypermineralized bone and to stop the progression of the disease. And then in another project that was awarded again, we demonstrated that this result is not reproduced, is not duplicated if we inhibit osteoclast function rather than osteoclast formation by using bisphosphonate. Specifically, we used zoledronic acid. And so from this previous grant, we came to the conclusions that it is possible to change the natural history, the evolution of FD lesions and of the disease by targeting osteoclasts and specifically osteoclast formation. However, as I said before, osteoclast function is very important to keep our skeleton healthy. And so inhibition, complete inhibition of osteoclast formation in the entire skeleton may have undesired effect, especially in growing kids. And so here comes the first point of the project that I'm going to show you today. The first question that we asked: is it possible to eliminate or at least to reduce osteoclast formation, osteoclast number selectively or preferentially within FD lesions, maybe by targeting molecules other than rank ligands? And we asked this question because in a parallel set of studies, again supported by the FD-MAS community, we observed that lesions, FD lesions that are particularly enriched in osteoclasts are characterized by higher level of expression of a molecule named brain-derived neurotrophic factor compared to lesions that have a lower number of osteoclasts. Now, brain-derived neurotrophic factor is produced within bone and bone marrow by different cell types, including osteoprogenitor cells and osteoblasts, and it is known to stimulate rank ligand secretion by osteoprogenitor cells. And it's known that it is involved in other skeletal diseases that are characterized by extensive bone resorption, bone destruction, such as, for instance, multiple myeloma. And so we first observed the expression of BDNF in paraffin-embedded tissue, paraffin-embedded bone biopsies from FD patients by using a method that we set up in the context of one of these projects that we, one of these grants that we received. But then we confirmed the expression of the molecule at both the gene transcription and protein levels in human and mouse fibrous dysplasia. And interestingly, we also observed that in mice, the level of expression of these molecules progressively increased with mouse aging. And so in order to test the potential role of BDNF in fibrous dysplasia lesions, we treated our mouse model with an anti-BDNF antibody twice a week for 3 weeks. And then we performed the radiographic and histological analysis. And here are the results. As expected, we observed a significant reduction in the levels of rank ligand in the FD lesions of treated mice, blue bar, compared to FD lesions of untreated mice, the gray bar. And we also observed the general reduction in osteoclastogenic activity, here highlighted by the specific red staining. And as you can see here, the level of staining was definitely lower in samples in lesions from mice that were treated with the anti-BDNF antibody compared to untreated mice. But obviously, we evaluated in detail change in osteoclast, and so we, by using histomorphometry, we could confirm a reduction, significant reduction in both in the number of osteoclasts, upper panel, and in the total area occupied by osteoclasts within FD lesions in the lower panel. In treated FD mice, blue bar, compared to untreated FD mice, white bar. And we also observed a reduction in the size of individual osteoclasts. This was very interesting because it's known that BDNF is also able to directly stimulate the fusion of osteoclast progenitors to generate large differentiated and functional cells. And it's interesting because it's known that somehow osteoclast size is related to osteoclast function. So we can say that we reduced the number and likely the function of osteoclasts within these lesions by using this approach. And this associated with clear changes in the histopathology of lesions of treated mice. In particular, we observed an increased amount of bone at the expenses of the fibrotic tissue. In the rest of the skeleton, osteoclastogenic activity continued with no major changes. And so I think that we can conclude that overall this part of the project, these results confirm the involvement of brain-derived neurotrophic factor in the abnormal process of osteoclastogenesis that occurs in fibrous dysplasia. And so maybe BDNF may be a target to modulate osteoclast formation within FD lesions. Obviously, these studies must be further expanded. We need to test different schedules. We need to test longer treatment. But I would like to remark here that we performed these studies in the last part of the COVID pandemic. So we have experienced a great delay in our work due to the restriction. Then in the second part of the project, we wanted to assess the importance of osteoclast and bone resorption in the clinical expression of fibrous dysplasia, meaning in bone pain, because we know that bone pain is an important problem in many FD-MAS patients. And so here we asked two different questions. The first one was a technical question. Is our mouse model a useful model to study bone pain in fibrous dysplasia? The second was an experimental question. If it is, what is the effect of inhibition of bone resorption on mouse bone pain? And so to address these points, we used and we are still using different well-established tests that evaluate the so-called pain-like behavior of mice, meaning the change in mouse behavior that reflects the presence of pain. We are using different types of tests. I don't have the time to go through all of them in detail today, so I will mention only the two most important tests that we are using, the burrowing test and the nesting test. Very briefly, the burrowing test evaluates the ability of mice to display food pellets that is placed in a tube. And the test consists in, actually, there is an habituation period that is required to reduce the variability. And then the test consists in two parts, a 2-hour and an overnight burrowing test. At the beginning of the experiment, tubes are filled with a few grams of food pellet, and the next morning, the remaining food is weighted. And so the ability of mice to display this food reflects the presence or not of pain. And the nesting test evaluates the ability of mice to build up a well-formed test. No habituation is needed here. So in essence, we give mice, provide mice with a few grams of shredded paper, and then we leave them work overnight. The next morning, we evaluated the nest and giving a score from 1 to 5 as previously reported, 1 for poorly formed nests and 5 for very well formed nests. And so here are the results. For the burrowing test, we evaluated 167 mice. So we evaluated the transgenic mice, females and males. Females are the red bars, males the blue bars, in comparison with wild type mice. We evaluated different mouse ages, and in essence, what we observed was a significant reduction in the burrowing activity, and therefore the presence of pain in transgenic mice. In female mice, we observed this reduction at all the different ages that we analyzed, whereas for male mice, the reduction was observed essentially from 5 months on. For the nesting test, we evaluated 240 mice, again, females and males transgenic mice in comparison with wild-type mice. And here we observed a significant reduction in the nesting activity of female mice, I would say of middle-aged female mice, 5-8 months age, and a reduction in nesting activity in older mice of both sex. And it's important to remark that neither for the burrowing nor for the nesting test, the results of the score, the evaluation was somehow related to the disease burden score, meaning to the extension of the disease that we evaluated on radio X-ray, by using X-ray. And this is important because this is consistent with what has been previously reported from human patients. And so this part support the validity of our model to study bone pain in fibrous dysplasia. And so, having assessed that we can use the model, we move to the most important part of this section of the project, meaning the evaluation of the effect of inhibition of bone resorption. And in this experiment, so we treated mice with the anti-rank ligand antibody, 4 doses or 6 doses, or with zoledronic acid, 2 doses, or with a combination of the 2 drugs. And control mice were treated with non-specific antibody, immunoglobulin, and with vehicle. And all mice were sacrificed 12 weeks after the beginning of the treatment. And so what we observe in these graphs, each dot represents a single mouse, and the behavior of these the individual mice was evaluated at the beginning of the experiment, T0, and at the end of the treatment, T12. And as you can see here, what we observed was an improvement in burrowing and nesting activity in mice that likely reflect a reduction in pain, in mice treated with the anti-rank ligand antibody, both treatments, whereas we did not observed major improvement in mice treated with zoledronic acid, and obviously neither in mice treated with vehicles and immunoglobulins as controls. But the most interesting and important results is that we observed the best, the greatest improvement in mice that received both the anti-rank ligand and the zoledronic acid. And so this suggests that this combined treatment may be important not only to prevent or to delay the disease rebound, but also to treat bone pain and fibrous dysplasia, to delay, the disease rebound after the anti-rank ligand antibody treatment. And so in conclusion, this part of the study, I think that these results may be taken as an important starting point for future studies on bone pain and fibrous dysplasia. For example, to assess the effect of different treatments on bone pain and musculoskeletal impairment, but also, and most important in my opinion, to investigate the pattern of innervation of FD lesions, meaning the pattern, the distribution of nerve fibers, the structures that are involved in the transmission of pain within FD lesions, both in basal conditions, meaning in the absence of treatment, and after different treatments. And this is actually part of the project of the last grants that we received. So we are performing now this study. And to conclude, I would like obviously to show the rest of the group, the Sapienza University group, which includes, besides myself and my co-PI Alessandro, young investigators, Biagio Palmisano, Giorgia Farinacci, Samantha Bensante, two new entries, Chiara and Alessia, too young to deserve a picture. And before concluding, I would like also to spend a few seconds on the importance of having this group. In particular, I would like to remind that we have been able to keep Biagio Palmisano, who is our senior collaborator, working with us for the last few years, thanks to the FDMAS grants. And so he has the opportunity to develop a great knowledge on fibrous dysplasia. He's been presenting this data at national and international meetings, has received awards, and he also has already received his own grants on fibrous dysplasia. And I wanted to remark this because I think that, preparing committed young research is a very important task and is a great achievement of this FDMAS grants that we received. And this was my last slide. So thank you very much for your attention.
Thank you, Mara. Those are two really fantastic projects. It's so nice to see that the FDMAS award money is being so fruitful. So thank you. If there's any questions, please ask. Naveen has a question. You're muted, Naveen. Are you able to unmute her, Tova?
Yeah, I'm trying. Oh, there we go.
There we go.
Yeah, I've done it. Yes, thanks, Mara, for a very exciting talk. I just have one question about combining an anti-rank ligand and zoledronate. Yeah, anti-rank ligand is so potent that what would you expect from adding zoledronate to it, and rather than giving it sequentially or consequent to the use of anti-rank ligand? I mean, what was the rationale of using both together?
Okay, actually these studies were based on a different rationale that was, we know that after anti-rank ligand withdrawal, there is a rebound of the disease.
Sure,
So
yeah.
We reason that if we provide bisphosphonates such as zoledronate during the treatment or in the last part of the treatment with the anti-rank ligand antibody, when we have the position of mineralized bone, maybe we have the incorporation of a great amount of zoledronic acid in the newly formed bone. And this molecule can be, this drug can be then released when we stop the treatment with the anti-venc ligand antibody in order to modulate, the rebound of the disease. This was the, and I hope that I've been clear, that I have addressed your question.
So
basically,
did you have a chance to do? Yes, but anti-ligand is so potent that would there be any cells for a bisphosphonate to work on, basically? Do you have any histology with just anti-RANK ligand and the combination of anti-RANK ligand and zoledronate? My question is, what is the added value? I do take your point about the theoretical basis of it, but what happens in, on a bone histology slide? What do you see comparing only anti-RANK ligand and anti-RANK ligand and zoledronate, if it is at the same sort of time span after you stop either one or the combination?
Okay, I can't talk about the histology yet, but we have X-ray analysis, and the X-ray analysis clearly shown that in mice that receive the combined treatment, there is a delay in the rebound of the disease. I mean, in the appearance of lesions. So it seems, at least at X-ray level, that it is working, and I'm sure that we will be able to confirm it by histology.
And has it got a more powerful effect on pain, for instance, do you think?
Oh, that's a very good point. This is something we are trying to understand. That's a great point. I mean, I personally don't have a clear explanation yet. So that's the reason why I think it's very important to evaluate the pattern of innervation within FT lesions after the treatment. I mean, we need to do that to better understand pain in fibrous dysplasia. We absolutely need.
Okay. Thank you very much. Thanks, Mike.
We've reached the hour. There's something in the chat from Cindy. She's been on the phone listening. Thank these researchers from the bottom of my heart, and hoping our community who are listening will continue with the quest for treatment and cure for FDMS and help support the FDMS for the June 10, 2023 ride. So I have to agree with that. Any last minute, and there's some other thank yous in here too, from other people, Dimitra, Mika, and Marilise Ekoff. So to the researchers and to the foundation. So I think though we'll end with that, and thank you so much, Mara. Thank you so much, Ted, Tova, Adrian, and Lily for all the work that you've done, and really great progress, and look forward to the future.
Thanks, everybody. Thanks so much.
Thank you very much.
Thank you, everyone. I just want to, before
Bye.
everyone hops off, I just want to say, it's wonderful to have these regular research updates online for the last few years, but we are really excited this year to be hosting an in-person conference in September in the Silver Spring, Maryland. We hope many of you are able to join us. Saturday is a community day, so opportunity for clinical care and community connection. And then Sunday is the International Research Meeting. That's the first official convening of the International Consortium for FDMAS Research. We hope that there can be more time for research, discussion, and collaboration like this. We'll have a poster exhibit, and we invite everybody to submit a poster to share and display your work. As part of that poster exhibit, there will be a contest for students and emerging investigators to earn some funding to attend the meeting. And again, we really think this is a great opportunity to continue collaboration and discussion. Early bird pricing for that conference goes through June 1st, and we hope many of you will be able to come. Otherwise, again, thank you so much to Cindy and to Lily for your dedication and leadership. Thank you to Mike for your service and the leadership you've shown this organization and the community. Thank you Doctors Abel and Riminucci for focusing your work, talent, and time on this disease. We're very, very grateful. And Mike, is there anything else you wanted to add about
No.
the conference or? Great.
All right, thank you.
Thank you, everyone, and do be in touch if you have questions.
Thank
Bye.
you. Bye.
Everyone.
Bye-bye.
Bye-bye.
Bye.
Right.